The human body is incredibly complex. It consists of numerous systems, including the circulatory, nervous, digestive, and immune systems, each with intricate networks and interactions. For example, 1. Circulatory system. It involves the heart, blood vessels, and blood, which work together to transport oxygen, nutrients, and waste products. 2. Nervous system. This includes the brain, spinal cord, and peripheral nerves, coordinating sensory information and responses. 3. Digestive system. It breaks down food into nutrients for absorption and expels waste. 4. Immune system. It defends against pathogens and includes various types of cells and molecules working in concert. Each of these systems relies on complex biochemical processes and interactions, demonstrating a level of sophistication that scientists are still unraveling. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for life. Here's a basic overview of how it works. 1. Structure. DNA is structured as a double helix, consisting of 2 long strands coiled around each other. Each strand is made up of a backbone of sugar and phosphate groups, with nitrogenous bases, adenine, thymine, cytosine, and guanine, extending from this backbone. 2. Base pairing. The bases on one strand pair with complementary bases on the opposite strand. Adenine pairs with thymine, and cytosine pairs with guanine. This pairing is crucial for DNA replication and transcription. 3. Genetic code. The sequence of these bases encodes genetic information. Groups of 3 bases, called codons, specify particular amino acids, which are the building blocks of proteins. 4. Replication. When a cell divides, DNA replicates. The 2 strands of the double helix separate, and each serves as a template for a new strand, ensuring that each daughter cell receives an identical copy of the DNA. 5. Transcription and translation. DNA provides the template for making RNA, transcription. RNA then guides the synthesis of proteins, translation, which perform various functions in the cell. Overall, DNA directs the development, functioning, and reproduction of all living organisms by encoding the necessary instructions in its sequence. RNA, or ribonucleic acid, is a molecule similar to DNA, but with distinct roles in cellular processes. Here's an overview of its key functions and characteristics. 1. Structure. RNA is typically single-stranded, though it can form complex secondary structures. Its backbone is composed of ribose sugar and phosphate groups, and it contains 4 nitrogenous bases: adenine, A, uracil U, cytosine, C, and guanine, G. Unlike DNA, which has thymine, T, RNA has uracil. 2. Types of RNA. Messenger RNA, mRNA, carries genetic information from DNA to the ribosome, where it serves as a template for protein synthesis. Transfer RNA, tRNA, brings amino acids to the ribosome during protein synthesis, matching its anticodon with codons on the mRNA. Ribosomal RNA, rRNA, a key component of ribosomes, which are the cellular machines that synthesize proteins. 3. Functions. Transcription. RNA is synthesized from a DNA template in a process called transcription. This RNA then carries the genetic instructions necessary for protein synthesis. Translation. mRNA guides the assembly of amino acids into proteins at the ribosome, where tRNA brings the correct amino acids based on the mRNA sequence. Regulation. Some RNA molecules, such as microRNAs, miRNAs, and small interfering RNAs, siRNAs, play roles in regulating gene expression and maintaining cellular functions. RNA is essential for translating genetic information into functional proteins and regulating various cellular processes. Gene expression is the process by which the information encoded in a gene is used to produce a functional product, typically a protein. This process involves several key steps. 1. Transcription. The first step is the transcription of DNA into messenger RNA, mRNA. During transcription, an enzyme called RNA polymerase binds to the promoter region of the gene and synthesizes a complementary RNA strand based on the DNA sequence. This results in a primary RNA transcript, which includes both exons, coding regions, and introns, non-coding regions. 2. RNA processing. In eukaryotic cells, the primary RNA transcript undergoes processing to become mature mRNA. This includes splicing. Introns are removed and exons are joined together. 5' capping. A cap structure is added to the 5' end of the mRNA to protect it from degradation and assist in translation. Polyadenylation. A poly-A tail is added to the 3' end of the mRNA to enhance stability and facilitate export from the nucleus. 3. Translation. The mature mRNA is transported to the ribosome, where it is translated into a protein. During translation, ribosomes read the mRNA sequence in groups of 3 bases, codons, each specifying an amino acid. Transfer RNA, tRNA molecules bring the appropriate amino acids to the ribosome, where they are linked together to form a polypeptide chain. The 4. Protein folding and modification. Once synthesized, the polypeptide chain folds into its functional 3-dimensional structure. Proteins may also undergo various post-translational modifications, such as phosphorylation or glycosylation, which are crucial for their final function and activity. Gene expression can be regulated at multiple stages, including transcriptional regulation, RNA processing, and translational control, allowing cells to produce the right proteins in the right amounts and at the right times. Gene expression is regulated at several levels to ensure that genes are expressed in the right cell types at the right times and in appropriate amounts. Key mechanisms of gene regulation include 1. Transcriptional regulation. Promoters and enhancers. DNA sequences that control the initiation of transcription. Promoters are located near the start of a gene, while enhancers can be found upstream or downstream and can enhance the transcription of a gene from a distance. Transcription factors. Proteins that bind to specific DNA sequences, promoters, enhancers, to either activate or repress transcription. Examples include activators and repressors. 2. Epigenetic regulation. DNA methylation, addition of methyl groups to DNA, typically at cytosine bases. This can repress gene expression by preventing transcription machinery from accessing the DNA. Histone modification. Chemical modifications to histone proteins around which DNA is wrapped. These modifications can either tighten or loosen DNA packaging, affecting gene accessibility. 3. Post-transcriptional regulation. RNA splicing, the process by which introns are removed and exons are joined together. Alternative splicing can produce different protein variants from the same gene. mRNA stability and degradation. The lifespan of mRNA in the cytoplasm can be regulated by RNA-binding proteins and microRNAs, affecting how much protein is produced. 4. Translational regulation. Regulation of translation initiation. Proteins and small RNAs can influence the initiation of translation by binding to mRNA and affecting ribosome binding. Regulatory RNAs. MicroRNAs, miRNAs, and small interfering RNAs, siRNAs, can bind to mRNA and block its translation or lead to its degradation. 5. Post-translational regulation. Protein modifications. After synthesis, proteins can be modified by phosphorylation, glycosylation, or other chemical changes, affecting their activity, stability, and localization. Protein degradation. Proteins are tagged for degradation by ubiquitin, leading to their breakdown by proteasomes. These regulatory mechanisms ensure that gene expression is finely tuned in response to developmental cues, environmental changes, and cellular needs. Yes, genes can be silenced through various mechanisms, which effectively reduce or completely inhibit their expression. Key methods of gene silencing include 1. DNA methylation. The addition of methyl groups to cytosine residues in DNA, particularly in promoter regions, can prevent transcription factors and RNA polymerase from accessing the gene, thereby silencing it. 2. Histone modification. Chemical modifications to histones, such as methylation or acetylation, can lead to tighter DNA packaging, making the gene inaccessible for transcription and thus silencing it. 3. RNA interference, RNAi. Small RNA molecules, such as microRNAs, miRNAs, and small interfering RNAs, siRNAs, can bind to complementary mRNA sequences and either degrade the mRNA or block its translation, leading to reduced gene expression. 4. Chromatin remodeling. Changes in chromatin structure through proteins that alter the organization of DNA and histones can result in a more compact chromatin configuration that inhibits transcription. 5. Transcriptional repression. Repressor proteins can bind to specific DNA sequences near or within genes and block the activity of transcription machinery, leading to reduced gene expression. These silencing mechanisms are essential for regulating gene expression during development, cellular differentiation, and in response to environmental signals, and they play a role in maintaining cellular homeostasis. Yes, Genes can be silenced, which means their expression is reduced or completely turned off. Gene silencing can occur through several mechanisms. 1. DNA methylation. How it works. Methyl groups are added to cytosine bases in DNA, particularly in regions known as CpG islands, which are often located near gene promoters. This methylation can block the binding of transcription factors and other necessary proteins, preventing the gene from being transcribed. Effect. Silenced genes are often associated with tightly packed chromatin, making the DNA less accessible to the transcription machinery. 2. Histone modification. How it works. Histones are proteins around which DNA is wrapped. Modifications to these histones, such as deacetylation, removal of acetyl groups, or methylation, can alter how tightly or loosely the DNA is wrapped. Tight wrapping, heterochromatin, makes the DNA inaccessible to transcription factors, silencing gene expression. Effect. These modifications can create a more compact chromatin structure, leading to gene repression. 3. RNA interference, RNA. How it works. Small RNA molecules, such as microRNAs, mRNAs, and small interfering RNAs, siRNAs, can bind to complementary mRNA sequences, preventing them from being translated into proteins or leading to their degradation. Effect. This post-transcriptional gene silencing reduces or eliminates the production of the corresponding protein. 4. Transcriptional repression. How it works. Repressor proteins can bind to specific DNA sequences in the promoter or enhancer regions of a gene, blocking the assembly of the transcription machinery, or recruiting other proteins that modify chromatin to a repressive state. Effect. The gene is not transcribed into mRNA, effectively silencing it. 5. X chromosome inactivation. How it works. In female mammals, one of the two X chromosomes is randomly inactivated in each cell through a process called X inactivation. This silencing is mediated by a non-coding RNA called cyst, which coats the chromosome and leads to its condensation into a bar body. Effect. The genes on the inactivated X chromosome are mostly silenced, ensuring that females, like males, have one functional copy of the X chromosome. 6. Genomic imprinting. How it works. Certain genes are expressed in a parent of origin specific manner. For imprinted genes, one allele, from either the mother or father, is epigenetically silenced through mechanisms like DNA methylation and histone modifications. Effect. Only the allele from the other parent is expressed, while the silenced allele is not active. These mechanisms are vital for processes like development, cell differentiation, and maintaining cellular identity. And they also play roles in diseases like cancer when gene silencing goes awry. Epigenetics refers to changes in gene expression that do not involve alterations to the underlying DNA sequence. Instead, epigenetic changes affect how genes are read by cells and how they produce proteins. This is different from genetic changes, which involve mutations or alterations in the DNA sequence itself. Here's how epigenetics differs from traditional genetics. Number 1. Mechanisms. Epigenetics involves modifications to DNA or histones that regulate gene expression without altering the DNA sequence. Common mechanisms include DNA methylation, the addition of methyl groups to DNA, typically silencing gene expression. Histone modification, chemical changes to the histone proteins around which DNA is wrapped, affecting how tightly DNA is packaged and consequently its accessibility for transcription. Non-coding RNAs. Small RNAs that can regulate gene expression post-transcriptionally by binding to mRNA and preventing its translation or promoting its degradation. Genetics involves the sequence of DNA itself, including mutations, insertions, deletions, and rearrangements that can alter gene function. Epigenetics. Epigenetic modifications are generally reversible. For example, DNA methylation can be removed and histone modifications can be altered, leading to changes in gene expression. Genetics. Genetic mutations are usually permanent and are passed on to daughter cells during cell division. While some genetic changes can be corrected by mechanisms like DNA repair, many are irreversible. 3. Inheritance. Epigenetics. Some epigenetic changes can be passed from one generation to the next, but they are often reset during the development of the next generation. However, certain epigenetic marks, such as those involved in genomic imprinting, can be inherited. Genetics. Genetic changes are inherited according to Mendelian principles, with mutations and variations in the DNA sequence being passed directly from parents to offspring. 4. Response to environment. Epigenetics. Epigenetic changes are often responsive to environmental factors, such as diet, stress, toxins, and other external stimuli. For example, exposure to certain chemicals can lead to changes in DNA methylation patterns, affecting gene expression. Genetics. Genetic changes are not influenced by the environment in the same direct way. While environmental factors can cause mutations, e.g., UV radiation leading to DNA damage, these mutations occur in the DNA sequence, not in how the genes are expressed. Number 5. Role in disease. Epigenetics. Aberrant epigenetic modifications can lead to diseases, including cancer, neurological disorders, and metabolic conditions. For example, abnormal DNA methylation can silence tumor suppressor genes, contributing to cancer development. Genetics. Genetic mutations can lead to inherited diseases like cystic fibrosis, sickle cell anemia, or Huntington's disease. These are caused by specific changes in the DNA sequence. In summary, while traditional genetics focuses on the DNA sequence and its changes, epigenetics involves the regulation of gene expression through reversible and often environmentally influenced modifications that do not change the DNA sequence itself.